Hi, everyone. Welcome back to the second part of the challenge of DNA. If you are joining this talk and you did not watch the previous talk, which is the challenge of DNA part one, I would actually really recommend stopping this video and going and watching the video right before this one, just because I'm going to kind of just continue with that talk. And so you might be a little confused at what we're talking about. Also, if you don't have a background in chemistry or biology, I kind of break down a few of the terms and definitions that I'll be using regarding proteins and nucleotide bases and amino acids in the previous talk. And so that would also help you to have a good foundation for this talk. However, if you've already watched that talk, thanks for coming, and I hope you are just ready to kind of dive deeper into the challenge of DNA. So, our last talk, we talked about the possible scenarios for a route to life. And so, in that talk, we talked about what are the three possible naturalistic routes to life. So, we talked about random chance and how probable is it for life to just kind of come together, combined in that primordial ooze that Darwin mentions, just randomly, just happening to be close proximity and then being produced. Second is natural selection, and we talked about what is actually the definition of natural selection. What is natural selection actually propose as being able to do, and how would that work in relationship to a route to life? And so remember, natural selection can only make something be improved. It can only increase the survival based on whatever characteristics is best fit for the circumstances that organism is in. It cannot actually create something new. It can just determine kind of which characteristics should continue in a population. So the third possible scenario is chemical affinities and self-ordering, and that's the one that we're going to discuss in today's lecture. And then we're going to have a little bit of a bonus at the end of just what is another argument for the route to life. So, chemical affinities. So, this scenario is going to focus on the various self-organizational theories for the origin of information-bearing macromolecules. So, remember that we're talking about DNA and that four-letter alphabet and how it would be able to kind of self-assemble. So, the idea here is that maybe chemical attractions caused DNA's four-letter alphabet to self-assemble. So, there was kind of like these attractions, you can almost think about it like magnets that kind of came together and caused the DNA structure. Also, natural affinities between amino acids prompted them to link together by themselves to create protein. So remember we said amino acids are kind of like little train cars, and the whole train would be the protein. And so between this train car and this train car, there was a natural affinity that would kind of make them link together and connect. So this is what this theory, this scenario is kind of proposing. There's these chemical attractions in the with the nucleotide bases, and then the amino acids have natural affinities. So let's look at that. So protein self-assembling. So proteins, like we said, are composed of a long line of amino acids. So it's not just one and two cars. It's a lot of cars to make this protein train, as you can think about it. And so the idea is that there are forces of attraction between the amino acids that would cause them to line up the way they do. And then there would be an additional kind of attraction that would cause them to fold in the way that they would need to be folded so that they could function. And then remember that the different foldings determine their function, and then that helps keep the cells alive because each of the proteins have different purposes and tasks. Now, do we see this kind of self-assembling in nature? We actually do. So there's an example of this natural self-ordering in salt crystals. So in salt crystals, you have a sodium cation, sodium positive, then you have a chloride anion, chloride negative. So you have a positive and a negative, so two different ions, positive and negative, and then they have chemical attraction and they kind of form a pattern. So you can see here, so the cations and the anions, and so then you have positive, negative, and they kind of form like this little structure going from like left to right and then top to bottom. So positive, negative, into this 3D kind of box shape. It's very highly ordered and very compact. And so we just get a sequence of NaCl ions over and over again, just like we see in this image here. So this is widely known. This is how salt, the structure looks. So we see this in nature, not a problem. But would this be an explanation for protein self-assembling? So what about proteins and DNA? So as scientists did experiments, they found that amino acids didn't demonstrate these bonding affinities. So these type of attraction between these two ions, this type of attraction is not found within the amino acids. There are some very slight affinities, but it's a different type and it's a much weaker affinity. But they do not correlate to these affinities do not correlate to any known patterns of sequencing that we can find in functional proteins. So it's not that even if it is an affinity, it isn't something that is going to cause a pattern. So here, this is how we would have these amino acids here. So you've got this amino acid and the second amino acid, and then they're going to come together. So think about this as the two cars of the train, and then they come together and link. But this linkage is not the same type of bonding affinity that we would be thinking about in relation to like salt crystals. So it's not an affinity, it's a bonding that is happening here. So what if there were strong self-organizing properties? Maybe there is, but we just haven't seen it. But if this existed, we'd actually have a really major theoretical problem. Because what would happen is we would get merely a repeating sequence. So think about that back into our salt. It's NaCl, NaCl, NaCl. It's a repeating, consistent sequence. So with DNA, it's spelled out with our four bases, A, C, G, and T. And so if we wanted to think about it with that affinities, those attractions, so if you think about it with our salt, every time we have an Na, we have a Cl. Then we have an Na, we have a Cl. Then with the DNA, if every time you had an A, it was a G, then you'd have A, G, A, G, A, G. So we would just get a repetitive sequence, and that repetitive sequence does not produce a protein. So to get a protein, you actually have to have variety, diversity within that sequence. And so just thinking about like if it's a code and just 1, 2, 1, 2, 1, 2, 1, 2, that's not going to actually write anything, a program or anything of that nature. So self-organization, this attraction, wouldn't actually yield a genetic message, something that's unique. It would only create a repetitive mantra. And so this actually wouldn't be a helpful explanation. So what we are trying to get from our DNA sequence is we're trying to get information so that we can then create the amino acids and the proteins that we need for life. But we don't need just this regular pattern. We actually need information. So information that's in that DNA kind of code that will help write what we need to produce. But to convey information, you need irregularity in sequencing. So think about it if we were to open a book and let's say you saw the over and over again, you wouldn't really get any information and it would be a very boring book. So we don't see when we open a book the over and over again. You see the kind of repeated throughout the book, but you also have lots of other letters and words that are associated in that book. And so you have an irregular sequencing of letters, of words. And that's how we actually convey information. Now, it's not just that we have, random words on the page. It doesn't say something, for example, if we look at my sentence, instead you have an irregular sequencing of letters. It's not like we open it up and it says have instead sequencing you letters and of irregular. I'm not sure if I repeated any of those, but it's not that you just have to have, irregularity in the sequencing, but you also have to have a kind of structure that has to fall under. So to convey information, you have to kind of have a certain independent pattern or rules that you're kind of staying in, like a box. And so when we're talking about writing a book, you don't want the and the and, we want variety. But those words, those pieces of that variety has to kind of fall into the rules of vocabulary and grammar. When they fall into those rules, then we get a nice, pretty story. So DNA is the same. We have those four letters of the alphabet. We need them to be in a highly irregular pattern, not A, G, A, G, A, G. But at the same time, they need to conform to a functional requirement. So they can't just be, there's a very certain ordering of what will actually make an amino acid that will then lead to a working protein. So we need the correct arrangement. It's not A or ATAT. There's a very specific ordering for each of the amino acids. So we need the correct arrangement to make the specific amino acid we need, and then we need the amino acids in the right order being made as well. And then we'll have a working protein. So let's talk a little bit more about these chemical affinities. So there's kind of two types or two kinds of processes. So there's complexity and there's simple order. So in complexity, we have variability, irregularity, and unpredictability. So you can think about this as information. In simple order, you have repetitiveness, redundant structure, and this would be like self-organization. So the way you could think about this is complexity and simple order are kind of categorical opposites. You can't have both. So if you wanted to be simple order, then this is kind of your NaCl, NaCl, NaCl. It's a repetitive, a redundant structure, simple order that you can get from self-organization. But if you want complexity, say for example, a very long book, like Anna Karenina, for example, just to name one off the top of my head, then because there's variability, irregularity, unpredictability of what's going to come next, each word, then you get information. And so it's not always going to be a predictable pattern. Now, specified complexity is that irregularity that's specified by a set of functional requirements. So we need the complexity, that variability, irregularity, and unpredictability, but we need it to be specified. So remember I said that you need it to be irregular, but it needs to be kind of in a framework, like the book. Like we can have all the words all over the page, but if they're not within the framework of grammar and structure rules, then it's not going to be very helpful. So we need specified complexity, so that irregularity that's specified by a set of functional requirements. And so this is going to be recognized as information. And then what kind of controls that and determines that specified complexity is the result of a mind. So DNA needs specified complex information to create all the different proteins needed for a living cell to function. So it needs to have the irregularity of the nucleotide bases, but they also need to be in a specific order that will produce the amino acids that are needed for the specific proteins that the cell is looking to have. So what about the structure of DNA? So this is if we were just looking at, what DNA leads to, but let's just look at DNA itself. So the structure of DNA depends on certain bonds that are caused by chemical attractions. So if we look here, there are hydrogen bonds and bonds between the sugar and phosphate molecules that form the two twisting backbones of the DNA molecule. So to get these little parts to kind of go around here, there are hydrogen bonds here in this sugar phosphate backbone. And then also where our bases match up here, so one base to another, and remember A and T always go together and G and C always go together in DNA. then we have hydrogen bonds. So you have hydrogen bonds kind of across here and then on this backbone. However, there are no chemical bonds between the nucleotide bases. So A, G, T, and C. So what that means is if we look at just one side, so if we look at just one side of this backbone, so this green cytosine and this red adenine, there's no chemical bonds between these two. So if we're talking about one side of the backbone, there is a hydrogen bond between this guy and the one he's attached to on the other backbone, but not between this guy and the guy next to him on the backbone, is what this is saying. So these letters do not interact chemically with each other in any significant way. Really, they're actually totally interchangeable. So who's going in each one of these spots is interchangeable. Each base can act with equal facility at any site along the DNA backbone. So what that means here is here you see it's green, red, yellow, so cytosine, adenine, thymine, but it could be, this could be a C, a G, a T, or an A. Like this little spot of where it's placed is interchangeable. There's nothing that determines which one this is going to be based on what's next to it. So it's not like, oh, because there's an adenine here, then there's a cytosine, adenine. Oh, wait, no, this should be, no. Like what comes before it in the line doesn't determine what comes next. So another way that you can kind of think about this, since that might be a little kind of hard to kind of process, is I want you to imagine a metal chalkboard, kind of like the one I have in this picture. So you might have had these like these little boards and you have like little magnetic letters that you can put on there. So we've got one of these little magnetic chalkboards and we have the letters, and I rearranged the letters on the board to spell information. So there are magnetic affinities here because this is what keeps these letters onto the chalkboard. So there's an attraction between the letter and the chalkboard, right? So, but the magnetic force is the same. If I put an A, a B, a C, or a D, they all have that same force to the chalkboard. So they're interchangeable. If I'm writing the word information, I could have put an I first, an A first, an F first, or an M first, because that magnetic force is the same for each letter. There's no magnetic force also between the letters. And so this is what I was talking about. So in the spots on the DNA, where it was, an adenine and a cytosine, what comes first, it could be any one of them. In that first spot, because of how it binds, it all binds the same. They all have the same type of way of attaching to the sugar phosphate backbone. So just like how all the letters have the same way of attaching to the metal chalkboard. So if I go back to the previous picture, this is what I'm saying with here. So this would be like the chalkboard, and each of these would be the letters. And so all of these have the same magnetic force here. And just like on this little magnetic chalkboard, these letters do not have a magnetic attraction to each other. So when we put them on the board, they're not, like going really, like pulling to each other and trying to be right next to each other. There's no magnetic attraction between the letters themselves. This is also the same of what I was saying with the backbone on the DNA. There's no attraction between the bases on the same backbone. The only attraction is with the base to the backbone. So to the of the letter to it. So it's the same kind of idea here that we get. So with that being said, if I now have the word information, where is that going to come from? So how did the word information arise on the metal chalkboard? It's because intelligence comes from outside the system. So all the letters could have been in that first spot, but I decided it was an I, and then an N, and then an F. And I, being this outside intelligent force, was coming in and determining that ordering. So it was not chemistry nor physics that arranged the letters this way. It's the exact same case in DNA and the nucleotide bases. That it's not chemistry or some chemical affinities or physics that determines the orders. It actually comes from outside the system, intelligence. So this is why the chemical affinity scenario is not a good explanation for DNA and the information that is provided in DNA. But also just remember that DNA provides some, but not all of the information that's needed to build a new organism. So if we want a new organism, we not only need DNA to build proteins, but we also need lots of other things. We need cells, we need tissues, we need organs, we need the overall body plans. So there's lots of different things that are needed and lots of information that is needed within um, the organism to be able to just exist and to live. So this is why these naturalistic routes to life just don't make sense because they do not explain where the information is coming from. And that's the biggest issue. Where is this information coming from? So now I want to kind of change gears just a little bit and talk about where some other information came from. So we've been looking at it from kind of a microscopic level, but there's another kind of historic argument of where all this information came from. So if this were true, if, say, natural selection or these chemical affinities and self-assembling was the way, the other thing that Darwin proposed was that we would be able to kind of predict the kind of transition and flow of life. So he was trying to say, where did life begin? And that's what we just focused on at the first part of this lecture, the previous talk video, and then how we just concluded that part. So now we talked about it from a very beginning of the atomic level of how life was kind of created, and is that possible? But we also wanted to say, okay, if that were all possible, how did Darwin kind of continue? And so he kind of said, okay, with a pyramidal soup, and then how everything kind of self-assembled, and then natural selection, and then we would slowly get to animal life and then humans. And so he had also his theory of natural selection and evolution. So here he was able to do this with an evolutionary tree. So this is just kind of a picture or a diagram that showed his thoughts of how this would proceed from the beginning of the first of life all the way until we have all of life. And so this tree illustrates his theory that all living creatures had a common ancestor. So if we look at the bottom of the tree, this would be the beginning, and then we would progress upward into this tree of life. And so all of life would start here at this common ancestor, and then natural selection would slowly drive the eventual development of the countless organisms that we see in the modern world. And so he believed that if a population was exposed to one set of conditions, so we have a group, they have one set of conditions, and then another part of the population experiences another condition. So say we have a group, one group is separated here, one group separated here, and they experience different conditions. Then natural selection would modify the two populations in different ways. So maybe one is in a really hot, arid environment, one's in a cold, wet environment, then they would kind of be modified. And over time, one species could produce several varieties, and these varieties would just continue to diverge and become separate species. So this one would then become two, and over time, they would end up being completely different. And so that would be kind of like you could see the idea here. So we had a group, one would go this way, one would go this way, and then they would slowly change based on the conditions and adapt, evolve. And then this group would slowly change, adapt, and evolve, and just continue until we get these branchings of this tree. So a key aspect of his theory is that understanding of natural selection. But we need, like we had talked about before, we need to really think about, well, what is natural selection? So natural selection, the way that Darwin defined this, is that this process would be a very slow modification. So what would happen is we would be slightly changing different characteristics and having different variations. So it's very slow, and we're just accumulating very slight successive favorable variations. And what it means by favorable just depends on the conditions. So what would ever be best in the conditions for survival in that population. And so the thing is here, you're not going to have any big, great sudden modifications. Natural selection just says it's a slow, gradual change of one characteristic that's more favorable in a population, kind of becoming the main, the majority, the dominant characteristic, and then that would continue and that then would continue on. And so it takes lots of generations and lots of time. However, it's very interesting to note that during Darwin's time, the fossil record did actually not support his theory. So even though he presented this theory, he really hoped that the fossil discoveries later would kind of vindicate his theory. So he said, "Here's what I think. Right now, I don't have the fossil records to prove it." But he was hoping with time and more discoveries in the fossil record that he would be shown to be shown correct. That his theory would be proven right. Well, actually, let's look at what the fossil record actually supports. And so what we see is that there is this kind of a long line and a point where a bunch of stuff happens, which is very similar to him. There's a long line and there's a point where kind of some stuff happens for him, only two things. But at this point, which is called the Cambrian period, a lot of things happen. So the Cambrian era is a geological period that began a little more than 540 million years ago. And so this event, during this era, during the Cambrian period, there was an event called the Cambrian explosion. It's also called the biological Big Bang. And the reason it's called the biological Big Bang is because, boom, all of a sudden, you have a appearance of most of the major animal phyla that are still alive today, as well as some that are now extinct. And so what that means is you kind of are going along with some of the same thing, and then boom, you have a lot of variety in the body plans that were seen in the fossil record. So it wasn't just slow, but it was like a bunch of different things kind of popping out all at the same time. So uh, before this time around this period, there were some jellyfish, some sponges, and some worms. But then after the Cambrian explosion, we see my apologies, representatives from the arthropods. So arthropods are like what we think of as modern day insects and crabs. The echinoderms, which are like modern day starfish and sea urchins. The chordats, which include modern day vertebrates, which actually the chordats lead as we continue down the line into mammals. So at this time, all of the major animal phyla appeared. And so when we say animal phyla, like this is our big grouping. All of the big groups are there. So no new groups are kind of appearing. So you might say, well, what do you mean by suddenly? You could kind of see, okay, there's some time, and we're kind of looking at time a little bit here, but maybe it's hard for you to think, okay, 540 million years ago, like what does that look like? So I want to kind of give you an example. And I apologize for anybody who is not an American football fan, but you can think about this as a soccer pitch or a rugby field or an Aussie rules field or whatever sport you would like. But I put a picture of a football field on here so you could kind of imagine it. So I want you to imagine yourself on the goal line. So we're right here on the goal line, if you can see my arrow. And so on the goal line here represents the very first fossil. And so this is a microscopic single-celled organism. So in our picture here, this would be at the beginning. Right here, this is where we're starting in both of our pictures. All right, so represents the first fossil right here. And now we start going down the field. So we're going down the field, we pass the 20-yard line, we keep going, still just that fossil. 40-yard line, still nothing. Hit midfield, still just microscopic single-celled organism, same little guy. And now we're coming and we're starting to approach the other goal line. and we're getting closer and we're getting closer. We still only see that microscopic single cell guy, and then we come right about here. So all of this time, we've seen pretty much almost the entire length of the football field, if that's our time period, our 540 million years. Or sorry, the entire time of that we have until 540 years, but my apologies. Um, that whole time, we just have a microscopic single-celled organism. And then right when we get up to this point, to the 16-yard line, we start to see some sponges, some jellyfish, and some worms. And then we take one step. So think about it as the 16-yard line, then one more step, and that's about the 540 million years. When we take that one extra step, then all other forms of animal life suddenly appear. So if we look at it from our timeline of the universe, of once we have kind of more life producing chemicals and things, and once the earth is kind of in a ability to sustain life, from that time point here on the goal line all the way down, we just have microscopic single-celled organisms. And then at 16 yards, we see the sponges and the jellyfish and the little worms. And then in one single step, we have this explosion. So that's what this means here. So here we start off at the goal line, and as we're walking, still the same thing, still the same thing. Then this would be the 16 yard line, and then with just kind of one step, which this should probably be shrunk up a little bit to be better visual. But at one step, this is where you get the explosion of all the little different animal phyla. So here's a picture of the two to compare. So on the left here is Darwin's Tree of Life, and on the right is the current kind of Cambrian explosion evolutionary bush, is what they call it. And so with the tree, it's this idea of branching based off of natural selection. And here it is where it expresses that at that time, all of the animal phyla were created. And so we don't have anything new being formed. All the major groups are existing, all at a single point. And so all of them can trace their time all the way back to the Cambrian explosion. Whereas here, only two of them can, not every single one of them. Now, some arguments that have been made in favor for Darwin's Tree of Life is, well, what if the fossil record is still incomplete? So maybe we're just have an incomplete picture like the bottom right here, and we don't really have the full picture. Well, the thing is, we have millions and millions of fossils that have already been dug up. And so if there was this missing fossil that was really going to like blow everything out and we're really going to see the full picture, it would have already happened. And it hasn't happened after all this time. So it's very improbable that there's something we're missing. That there's some group of fossils that we just haven't found yet. There's a lot of sedimentary rock from before the Cambrian explosion that has preserved ancestors that we've been able to look. So it's not like, oh, we haven't found rock that old. No, we do have rock before the Cambrian, and we have a lot of different sources, and we've been able to kind of get a good picture of what it would all look like at that time. So it's not like we're missing anything because we have such a wide and broad source of fossils from before the Cambrian explosion. So the other thing is the Cambrian explosion is just too big to be masked by flaws in the fossil record. So what they're trying to say is that we're missing something, but there's no explanation with this idea of missing something that will explain why all of this is popping up. It's not that all of a sudden just two pop up. All of the animal phyla pop up, and we don't see any hints of them before. And so it's not like we're missing one or two. We would be missing all of them in the fossil record. So to say it's incomplete is to say you're missing a really, really big explosion, and you're seeing no even hints of evidence of it before that explosion in time, which is what natural selection would say, is that there should be some small changes, kind of those transitionary fossils that should be in the record, kind of leading up to it, showing you how they were coming. And so it shouldn't just be a big, so remember I said natural selection, you don't have these big giant leaps. You should have a bunch of small little changes that we should see in the fossil record, but we don't. We see one kind and then we see all of them. And so we don't really have that gradual process in the fossil record for any of the animal phyla, more or less even one or two. And so that's what it means by it's just too big to kind of claim that the fossil record is incomplete. Now, some people would say, well, what if the organisms that existed prior to the biological Big Bang were just too small, when their bodies were kind of soft, and so they didn't really leave a trace in the fossil record. So maybe we had the beginning and we have the major phyla, but the intermediate guys, just the way that they were, they wouldn't be caught. Well, it's interesting because we have microfossils of bacteria and rocks dating back more than 3 billion years. So if we have microfossils of bacteria, then anything that should, in this natural selection progression, be the same size as microfossils or be a little bit bigger but still small should be able to be seen. So this argument of them being too small, we have really, really small ones already in our fossil record that we can find that date before the Cambrian explosion. Now, what about them being soft? We also have soft body organisms from before the Cambrian. So remember I said that you had some little worms and those kind of guys before the Cambrian. So we have evidence of soft ones before, so it shows that that can't be an explanation. And we also have soft body animals during the Cambrian. So it being a soft body and not being able to kind of be found in the fossil record also isn't really supported because we have it before and during the Cambrian explosion, we have those. So why we're not seeing those fossils in the fossil record, there's not an explanation. It's not because we haven't found them. And it's not because, oh, they wouldn't have been preserved. It's because they don't exist. Those animals didn't exist at that time. They only existed once the Cambrian explosion happened. But the Cambrian explosion does not support Darwinian evolution and natural selection. And so that is the controversy there. So I'm going to leave you with that information, just a little bit about the Cambrian explosion. And in my next talk, we're going to dive a little bit more into evolution, and we're going to look at humans and chimps. And what is the idea of where did human life, Homo sapiens, come from? Did we evolve from chimps, or is it something else? So I look forward to having you at my next talk. Here are the references, just a few regarding the second part of the challenge of DNA.